Okay, hey guys, it's Justin. I'm back for more in your microbiology lab. Having fun doing all the experiments for you and being able to demonstrate them for you. So today we're going to do a serial dilution, and this allows us to do what's called a standard plate count by diluting from a stock culture and diluting it out to very few cells. We can then be able to count how many colonies form on a plate, and then for a somewhat accurate estimation of what we started with. So in other words, we're going to take a stock culture and dilute it out, put it onto plates, and then be able to count the colonies, and that'll give us an idea of how much bacteria is in the original stock culture. So you can read more about it in your lab manual, and I'm just here to kind of show you the technique in doing this. So what I have here, I have everything I need within reach. So I have my pipetters, I have my tubes, I have my pipette tips, and in each of these tubes here then I have a diluent, and in this case it's just sterilized water. The first two tubes have 990 microliters of water, and the last three tubes have 900 microliters of water. So for the first tube then I'm going to take 10 microliters of my stock culture and add it to 990 microliters of water. I'm going to do the same thing for the second tube, but I'm going to take out of the tube that I just diluted, and then so on and so forth till we're left with a very dilute solution of cells, and then we're going to plate the last four dilutions on to TSA plates, and then grow them in the incubator overnight, and then count how many colonies are resulting on those plates. Then that'll give us an idea of how many cells we started with in the original culture. So let's get started. I'm going to first take 10 microliters of the stock culture and insert it into our first tube. Okay, so let's grab these tips. Actually, for this smaller pipetter, I'm using the 2 to 20 microliter pipetter, and it's set at 10 microliters. So I'm going to flip this open and flip that open, and I'm going to pipette up and down a little bit just to mix it. Okay, and I'm going to disperse it kind of at an angle here on the side of the tube just to make sure it gets into the solution there. Close it, and I'm going to exchange tips between each tube so we don't cross-contaminate. And I'm just going to flick this to mix it. Okay, on to the next one. So 10 microliters out of the first tube that I just did into the second tube. I'm just going to go ahead and pick this up right here. So 10 microliters out of here into my second tube. Pipette up and down a little bit to make sure it gets in there. Okay, now I'm done with this tube, so I'm going to go ahead and set it up there so I don't lose track of where I'm at. I'm going to mix this up really good. Okay, now we did 10 microliters from these first two tubes. Now we're going to go 100 microliters into the rest of these. Okay, so I'm going to put this one away and grab my 100 microliter pipetter, which I should have already had set, but I didn't, so excuse me a second. Let me get this going to 100 microliters, and there we go. Okay, lock that in place there. And I'm going to need these bigger tips I'm going to set these aside. Okay, so now I'm going to go 100 microliters from my second tube into my third tube, which has only 900 microliters of the water. Going to pipette up and down to mix it well. And into my third tube. All right, done with my second tube. I'll put that up there. Mix this. All right, flip that open. Get my fourth tube. Get a new tip. Mix a little more. And into my fourth tube. I'm done with my third tube. Put it up there. Mix this one. Right, get my fifth tube. And this is my last serial dilution here into my fifth tube. Close them up and mix this one really well. So now what we're going to do is take a sample from each of these tubes and put them onto plates. So I've already labeled my plates A, B, C, and D, and I'm just worried about my second, third, fourth, and fifth dilutions. So those will be an actual, a volume portion of our original sample. So you'll see in your lab manual what each tube corresponds to volume-wise to your original sample. So I'm just going to take 100 microliters of each of these and plate them. So tube 2 will be A. Set that aside here so you can see it. I'm going to grab my 100 microliter pipetter. It's already set at 100 microliters. I'm going to remove the lid and take a sample from tube 2 and put it on plate A. Put it right in the middle. I'm going to cover the lid while I get our cell spreaders. So these are sterilized single-use cell spreaders. And I'm going to use a new one for each of these. So I got my sample in the middle there. And now I'm just going to go back and forth as I spin my plate and just evenly disperse it across the whole surface and really spread those cells out. Alright, so that one's good. So onto my tube 3, onto plate B. So grab 100 microliters out of tube 3. Mix it a little more. Okay. So tube 3 onto plate B. And spread these out. Okay, it looks good. Set that one aside. Now tube 4 onto plate C. Okay, mix it a little better there. Disperse right in the middle. And spread. Okay, good. And set that aside, and then onto the last one, our most diluted sample.